Hello students, how are you all? Welcome to One Page Biology. So in today's video, I will be talking about another very important part which is called as translation part 2. Now dear students, in the previous video I had covered translation part 1, and if you have not seen the video, please make sure you watch that video. Now when we talk about translation, what do we mean by translation? Translation simply means the information is converted from one form to another. But here in terms of a cell, what do we mean by translation? Here the information which is actually present in the RNA, and you may wonder that what is this information? So this information is mainly of the proteins. So this information which is present in the RNA will be converted in another form, that is nothing but the physical form of proteins, and that is nothing but translation, right? So in simple words, if you have to say that the information initially is present in the DNA, which then later on goes to the mRNA. If you remember, mRNA is nothing but messenger RNA, and the process by which the mRNA is formed from the DNA, it is called as transcription. And once the mRNA is formed, then from the mRNA, along with the ribosomes and the tRNA as well as the rRNA, this information which is present in the mRNA will be actually converted into the proteins, and this entire process of formation of proteins is called as translation. And together transcription and translation is known as central dogma, right? So let us understand, dear students, how the translation mechanism occurs, or how does this process occur. So now dear students, as you can see on the screen, that this process of translation is mainly divided into 3 steps. And what are these 3 steps? These are nothing but initiation, elongation and termination. Now you may wonder that what exactly happens in each of these steps. So let us try and understand one by one what exactly is initiation, what is elongation and what is termination. Now when we talk of initiation, now dear students, initiation means the beginning, right? So the process of translation is going to start in the first step. What exactly happens in the beginning or the initiation? In the beginning, simply what happens is there is a mRNA which we all know, which is in the direction of 5' to 3', and on the mRNA we know that the first codon which is present on the mRNA is known as the start codon, and this last codon which is present on the mRNA is known as the stop codon. Now in the first step, that is initiation, what is going to happen is initially the tRNA, which is also called as the transfer RNA. This transfer RNA actually binds with the amino acid, right? So what is this step called? This is known as activation of amino acid, or sometimes it is also known as activation or the charging of tRNA. It is also called as charging of tRNA, right? So the amino acid is going to get attached to the tRNA, and how it is going to get attached? It is going to get attached with the help of an enzyme which is known as amino acyl tRNA synthetase. This enzyme, by using the ATP we all know that it is the energy rich molecule. So along with the ATP, this enzyme combines the amino acid to the tRNA. Now this is the first step of translation. So once the tRNA is activated, it is now ready to transfer that amino acid to the mRNA. This is the structure of tRNA. Now dear students, I will be making a separate video to explain you all the structure of tRNA. It is also known as clover leaf model, right? But I will be explaining you it in the another video. So let's try and understand. Here the amino acid is getting bind at this particular site, that is the 3' end of tRNA, along with the help of ATP, right? So ATP is getting converted to AMP, and there are 2 phosphates which are getting released. Once the amino acid has been attached to the tRNA, now the tRNA is ready, or you can say such a tRNA is called as the charged tRNA. Now in the next step, dear students, what going to happen once the first tRNA is ready? Now remember, now the first tRNA is going to go and attach to the first codon of the mRNA. And what is the first codon? The first codon is always AUG. Now the first codon is always AUG, which is called as the start codon. Now the first codon AUG always contains the information of the amino acid which is known as methionine, right? So as per the genetic code, we all know that there are 64 codons, and in those 64 codons, the 3 codons are stop codons which do not code for any amino acid. So the remaining 61 codon, which are basically the sense codons, they have the information of amino acid, and in that the first codon, which is known as AUG, is the start codon, and start codon has the information of nothing but the amino acid which is called as methionine, right? So as you can see in the diagram, that the first tRNA is actually coming in by taking the amino acid, that is MHT, which stands for methionine, and this first tRNA is going to go and attach to the first codon, which is known as start codon, that is AUG. So now see, initially what is going to happen is, on the mRNA, first the small subunit of RNA comes and attaches to the start codon, and then the tRNA is going to come by bringing the first amino acid, that is the methionine, followed by the another large subunit of the ribosome. So as you can see on the screen, that there is a large subunit also which is going to get attached to the mRNA. Now how the tRNA actually binds to the start codon? Now tRNA has got anticodons, my dear students. So AUG has got a complementary anticodon which is UAC. And once the tRNA attaches to the mRNA, then the large subunit is going to come and going to attach. So now this set which is formed, this is known as an initiation complex. Now the process of translation has started, dear students. Okay, so now what is going to happen? Now in the next step, okay, now please remember, dear students, in case of eukaryotes, the first amino acid is methionine, but in case of prokaryotes, what do we mean by prokaryotes? Prokaryotes include the bacteria. So in case of bacteria, always the first amino acid is F-methionine, wherein F stands for formyl methionine. Now once the initiation complex has been formed, now the process of translation will continue. That is, now the tRNA will keep on coming on the mRNA, and basically the new amino acid will keep on coming in with the help of tRNA. Now see what exactly happens. Now dear students, you have to understand one thing over here, that now in case of ribosomes, can you all see there are 3 different sites inside the ribosomes which have been mentioned. One is called as the E site, the another is P site, and another is A site. Now what is E site exactly? E site stands for exit site, P site stands for peptidyl site, and A site stands for amino acid site. Right. So now please remember, dear students, as you can see that on the P site there is a tRNA. Now why have they shown on the tRNA? Because always remember, the tRNA comes, the first tRNA comes at the P site, which is the peptidyl site. Right. Now as soon as the first tRNA has come, now what will happen? The second tRNA will also come, which is at the A site, and A site is known as the amino acid site. Now once the second tRNA comes, there will be a formation of a bond between the first amino acid and the second amino acid. Now can you guess what kind of bond will be present between the amino acids? Yes, dear students, you have guessed it right, it is peptide bond. So now the peptide bond will be formed between first and second amino acid, and at the same time what will happen? Once the bond is formed, now the ribosomes will slightly shift ahead. Now as it has been as you can see on the screen, the ribosomes will slightly move ahead. As a result of it, the original A site is now going to get converted into P site, and a new A site is created. That is very important, dear students. Let me repeat it again. The new A site will be created, and the earlier A site has converted into P site, whereas now there is a E site which is created, dear students, and the first tRNA which was present, it is going to exit the ribosome from the E site, which is known as the exit site, right? So this movement of ribosomes on the mRNA is called as translocation, and like this, now dear students, each tRNA will keep on coming in, a new tRNA will keep on coming in with a new amino acid, and that's how a chain of polypeptide will be formed. Now this will happen, dear students, this will keep on happening. As you can see on the screen, the ribosomes will keep on moving on the mRNA, and a chain of polypeptide will be formed. Now this will keep on happening till the point a stop codon has been reached. Now once finally the ribosomes reach to a stop codon. Now what is a stop codon? Now there are 3 stop codons as per the genetic code, which are nothing but UAA, UAG or UGA. Now once the ribosomes actually reach to the stop codons, now please remember there are certain factors which are released in the cell. These are known as release factors. These factors get released in the cell, and once these factors are released, basically the polypeptide chain which has been formed, it is going to get released in the cell, as well as both the subunits of the ribosomes are going to get separated, and the mRNA is also going to get released in the cell. Finally, it is going to get degraded. That means the process of translation is over. That means the polypeptide chain has been now been made. It is ready to. So this was the translation process, dear students. If you have understood the process of translation, please like, share and subscribe to One Page Biology and share it with your friends as much as possible. I will be coming with more and more biology related videos for you all on One Page Biology. Till that time, take care, dear students. Thank you. Thanks a lot.